Hello and welcome dear viewers to the informative video on generic product development. And in this video we are going to have the understanding about the steps involved in generic product development. So generic product development may differ from applicant to applicant or from company to company. These steps or the product flow or the generic product development flow may change or may have variations from company to company, product to product also, and based on the product criticality. Whoever are working into the pharmaceutical field, these people should know how is the generic product is developed and what are the steps involved in generic product development. So I hope this video will help you out to understand the generic product development stages or steps. This video will give you the understanding and in-depth knowledge about the different stages of the formulation development till the commercialization stage. So let's start with the video. I am trying to make this video as short as possible. So be patient while studying or while viewing this video. See, generic product and reference product you might have heard. So generic product and reference product, we have to understand what is the difference. See, the reference product or the innovator product, sometimes it is also called as comparator product, is the product which is approved for the first time by the regulatory authorities. Generally, whenever there is development of NC, that is new chemical entity or new molecular entity that is formulated into the formulation and the NDA is filed. If you consider the US market, then it is called as NDA or it is referred as innovator or it is a reference product. So that reference product is applied to the regulatory authority based on the complete clinical preclinical studies, that is involving the bioequivalence studies and bioavailability studies. Bioequivalence studies means the bioequivalence between the different strains also and within the different formulations. And generic product is the product which is equivalent to the reference product or comparator product or RLD or the innovator product. And generic product is also known as ANDA or abbreviated new drug application if you work for the US market. The generic product is the product which is equal to the reference product considering its quality and the bioequivalence. So generic product should have pharmaceutical equivalence and the bioequivalence so that it can be given in place of reference product, reference listed product or innovator product or the comparator product. That means the generic product serves as a second product for the reference product. If reference product is not available in the market, the physicians can give the generic product in place of the reference product, or they can do interchanging for the reference product, and that is because the generic product is pharmaceutically equivalent and it is therapeutically equivalent to the reference product. So this is the generalized understanding about the generic product and the reference product. Generic product does not involve the complete preclinical and clinical studies. However, the generic product applicant does the bioequivalence studies with the innovator formulation and proves that the generic product is not significantly different or is the same replica of the innovator formulation or innovator product, and that is the basis of the generic product approval. See, The whenever generic product comes into the development, the market and regulatory authority for application requires to be understood, and it should be very clear. If the product is being developed for multiple markets or multiple countries, then the regulatory authority requirements of each of the countries should be taken into consideration. Then comes the infringing or non-infringing strategy for the development. If the generic applicants wants to develop a totally generic product, that means with infringing strategy or non-infringing strategy, whether the generic applicant is going to wait for the patents of the innovator to expire or not. So that should be clear from the first day. Then a reference product or RLD availability. If you are going to develop a product and you are not having RLD, or RLD is not there in the market, or there is very limited lots of RLDs available, then it may have, or it may come with the difficulties for the generic product development. So every time whenever the generic product is developed, the applicant and the people working on that product should have complete understanding about the market regulatory authority for the application, infringing or non-infringing type of formulation for the development, and reference product availability. Sometimes the reference product is not launched, but the applicants start developing the generic product. So that is also a case, and in that case, generalized compositions are studied, and if that is ER product, then different profiles of the ER formulations are made, so that whenever the RLD is launched, the available test product can be tested against the RLD. Then the generic product development start with the literature search, patent search, and the infringement analysis. Infringement analysis involves the study of the patents, what is claimed into the patents, and how these patents can be uh, infringed, or how these patents will not be infringed. So based on that, the infringement analysis is performed. Starting with the requirements for the generic product development are the API, raw materials, packaging materials, change part, toolings, and the technology. So whenever working on to the generic product, from initial we have to see whether there are the pharmacopoeial monographs available for API, excipients, and the products. If the product claims the USP, for example, paracetamol tablets USP or metformin tablets USP, that means there should be a monograph into the USP. And based on the basis of these monographs, this literature survey is done. Then patient information leaflet or product summary and reviews of the innovator products are taken into consideration, and these are required to be studied thoroughly, so that you will get the information about the reference product strengths, reference product indications, reference product toxicology, reference product information about the pharmacokinetics, pharmacodynamics, and the maximum daily dose of the innovator. That will help you to understand the generic product development. Then API DMF, drug master file, patents related to API and the patents related to product are required to be studied. Then also scientific literature available on the analysis, dissolution method, impurities, pharmacokinetics, and the pharmacodynamics of the API or reference product should be studied in detail. And this reference product study, literature study, patent search, and patent study will help you to develop the generic product. You will get complete and clear understanding about the reference product, and then you can develop the generic formulation. So literature search, patent search is the first stage or first step of the generic product development. Second stage comes as a license related work. So whenever you get the product for development, you have to have the licenses like test license to start working, then import license for API if API is available in the foreign countries. That time import license for API is required. Import license is required for the reference product, and these import license are the country specific. Then manufacturing license for manufacturing the product at commercial site, and BENOC, that is bioequivalence study NOC, no objection certificate from the regulatory bodies where these bioequivalence studies will be performed. So these are the these are the license related requirements. Then coming to the sourcing and evaluation of the sources. So API requirement will be there. First of all, we have to consider whether the API requirement is DMF grade API, CEP grade API, or monograph requirement or monograph grade API, or in-house testing based API can be used or cannot be used. So that should be clear from the first day. If you are going for the regulated markets, then you must have a DMF grade material. You can select the two or more sources for the same API so that you can have the better understanding about the API. Always we have to evaluate 2-3 lots of the API to have understanding about the uniformity in the API lots produced by or manufactured by the vendor. Then excipients and raw materials which are required for the formulation. These are taken from these are taken from the patient information leaflet or composition of the RLD. So from there you can understand that which excipients or raw materials are used. Then machine change parts. Machine change parts are required sometime for some specific products. Uh, the example is dosing disc for capsule formulation and punch tooling for the tablet formulation. Then packaging materials. What material is used by the reference product, the same material can be used for the generic product. And that packaging material, if it is understood that which material is used by the innovator, based on that you can understand the criticality of the stability or generation of the impurities. Then machines are also required if the product involves new technology. If the product involves use of spray drying or Worcester coating or hot melt extrusion or melt granulation or some newer technology like powder layering, that time for the generic product development the machines are required to be procured. So this is regarding the evaluation of the sources. Now coming to the API sourcing. API sourcing should be done from 2-3 vendors. DMF availability and compliance to the regulatory requirement is required to be checked. Then non-infringement statement or declaration is taken from the vendor. Nowadays the nitrosamine declaration is also taken from the vendor so that it can be understood that the vendor is providing nitrosamine free API, or it can be seen that whether the vendor is doing the nitrosamine risk assessment or not. Then particle size specification for the API from vendor and train data. Suppose API manufacturer has manufactured 50 batches, so the PSD data of those 50 batches can be taken. Similarly, bulk density, tab density data can be taken. Then polymorphic form statement. So the API might be present in 2 or 3 polymorphs or maybe amorphous. So that statement should be taken from the vendor that the material provided by specific vendor is which form or is it amorphous, or is it any specific form. Then impurity and physical properties specifications. These are taken from the API vendor. Generally, most of the things are available in the DMF, and if the open part is supplied by the vendor, you can get much of the information inside that open part of the DMF. Then stability and FD data is required to be studied and checked. Then API vendors packaging for the API is required to be studied. How that API is packed and provided. What are the storage requirement and what is the stability. Then analytical method for the various testing can be taken from that API vendor, and that will serve as a starting point for your method development. Source evaluation and lead time to provide the API. So always the API vendor should be asked the question that in how much time you can deliver the API. If we finalize your source, then how much time you will take for providing the API after giving the order or placing the purchase order. Then costing of the API is a very important thing, and costing should be evaluated thoroughly. Many of the times the APIs are infringing, sometimes these are non-infringing, sometimes some vendors have different costing, sometimes some vendors have different impurity profiling. So based on that, the costing negotiation should be done and costing should be finalized. Then coming to the reference product, raw material, packaging material, change part and toolings. So purchasing of the innovator formulation is required from the specific country for which country the generic formulation is being developed. The reference product is required for the development activities like matching dissolution and physical characterization, and also for the bioequivalence studies. Availability and requirement of the new excipients. If any new excipient is used, that excipient is required to be evaluated and should be sourced. Then raw materials like capsule shells for the capsule formulation and film coating compositions for the tablet formulations. Then tooling requirement. Tooling requirement and tooling development is done based on the reference formulation, and change part like dosing disc for capsules is done based on the powder characteristics of the generic formulation. Then in the same way, the packaging materials can be developed and packaging material can be finalized. Then API testing and evaluation. Now you have the test license and you have the API vendor finalized, you have selected the API vendor, and now that API is reached to your site. Now API evaluation and analytical method development is required to be started. Suppliers method can be used for this purpose as a starting point. Then for API, the particle size, bulk density, tab density, flow properties, microscopic studies, hygroscopicity, polymorphic form studies by XRD and DSC can be done. Then microscopic studies are done to evaluate the particle shape. Solubility studies for the API involves BCS solubility and the saturation solubility as per the ICH requirements or the country specific requirements. Then solubility in water and organic solvents is also required to be performed so that you can go for the non-aqueous granulation like example, or the aqueous granulation, or you can have the understanding that in which solvent the API can be dissolved. So that solubility study will guide you for the formulation development. Now force degradation and the stress study is required to be performed for the API. Then comes to the excipient compatibility. Whether any interaction is there between the API and the excipient, that is required to be studied based on the assay and the impurity testing. Then reference product testing and the evaluation. Now you have the import license, you have purchased the innovator, and now you are going to evaluate the RLD. So API form of the RLD, PSD of the RLD is required to be tested. It involves physical characterization, characterization of the critical excipients, like if the RLD composition contains the solubilizer, that time that excipient can be critical for the dissolution. So that excipient can be characterized or quantified. Then Q1, Q2, Q3 information is required to be generated. Complete analysis and dissolution studies are required to be done on the reference product. Then reference product is also studied for the impurities and their levels, to have the understanding that whether the innovator is using a stable API or whether the innovator itself is getting degraded in some conditions. Characterization of the innovator packs. So whatever the packaging material is there used by the innovator, that packaging material can be evaluated, and based on that the packaging material can be selected for the generic formulation. Deformulation, to understand the manufacturing method, excipient grades and the quantities. So evaluation of the RLD for disintegration pattern, for particle size shape, for disintegration mechanism or disintegrant activity, or it is also evaluated for the quantities. And based on this characterization, the QTPP and CQAs are designed for the test formulation. Then once you have all this information, you can go for the pre-formulation activities. Pre-formulation activities are product specific. Pre-formulation activities can be done at any point of the product development based on the requirement and the criticality. If you change the composition, then once you have to do the pre-formulation again, that is drug compatibility study. And generally the compatibility studies should be done on the final composition and with the final testing method. Then the formulation development starts. Initial trials are taken with some tentative composition, and samples are given for the analytical method development. And based on that, based on the CQA's requirement like dissolution, assay, CU and impurity profiling, the formulation is developed. The trial batches are manufactured by various techniques like direct compression, roller compaction, wet granulation, and many other techniques based on the reference product characterization and the CQA requirement. CQA are nothing but the critical quality attributes like assay, CU, dissolution, water content, and the impurity profile. Then stability data for formulation is required to be generated in various packs at different conditions, and these stability conditions are finalized based on the specific regulatory requirements like ICH requirement, US FDA requirement, EMA requirement, Brazil requirement, Zone 4B requirement. So based on that, these studies are designed. Then based on the prototype development, the prototype composition and the process is selected for the further formula and process development. Then prototype formulation involves the selection of prototype composition, quantitative composition and qualitative composition, manufacturing process and container closure system, that is packaging system for the generic product. See, this prototype formulation is to be fixed for the pilot bioequivalence studies. If you are manufacturing or if you are going to manufacture the exhibit batches, that time the prototype formulation should be evaluated very critically. The finalization of the prototype is based on to the criteria that it meets the QTPP and CQA. Dissolution method development and finalization of the parameters. And once you have the prototype formulation, you have to do dissolution method development, you can challenge the formulation by different dissolutions, like changing the media, changing the RPM, so that the formulation behavior should be understood. Then reproducibility trials are taken for the prototype formulation. Once you have finalized the prototype composition and the process, then you should check the reproducibility. Then pilot bioequivalence study. This pilot bioequivalence study is done along with the reference product to study whether the prototype composition will be bioequivalent to the RLD or not. Many of the times, one or two prototypes are taken and parallel or crossover studies are done. The advantage of taking 2-3 prototypes is that you will have the IV-IVC correlation data. For this, generally the prototype 1 with matching profile or slightly lower profile, another prototype with matching profile to the dissolution of innovator, and the third is faster. So by this profiling of the dissolution of test formulation with respect to the reference formulation, the bioequivalence study is done, and the outcome of the bioequivalence study will give you the clear understanding about the dissolution of the test product, whether I have to keep the dissolution faster than the innovator or I should match the dissolution to the reference product. Then the risk assessment. See, this risk assessment part is not fixed and it can be done at different stages based on the formulation requirement. So risk assessment is done for the drug substance, raw materials, and the manufacturing process. Also, risk assessment is done for the composition and quantitative composition. So based on that risk assessment, critical material attributes are identified for drug substance and the raw materials, and critical process parameters are identified for the process. So stages of the risk assessment are initial risk assessment. Then once you finalize the risk, then you have to study the factors affecting to that risk. That involves ongoing risk assessment involving the optimization studies. And formulation optimization studies may be done with DOE trials using software and OFAT trials, one factor at a time type of trials. Then based on the outcome of these trials, risk is required to be updated and you can finalize the control strategy. This control strategy can be finalized after the scale up and registration batches and also updated after the validation batches. And based on this control strategy, the life cycle approach is taken. Then coming to the scale up and tech transfer. Once you have finalized composition, once you have the prototype formula and process product with matching the RLD in bioequivalence study, that means you have cleared the bioequivalence study, you have the stability data, you have the robust process, then the scale up and tech transfer activities can be initiated. So for this scale up and tech transfer activity, finalization of composition and process is required. Then finalization of the specifications for API, raw material, packaging materials are required. Finalization of the specifications for initial release in process materials and the stability of the formulation is required. Then scale up challenges and protocols are finalized so that in scale up studies you will have the written document for performing the challenges. For example, machine speed challenges for some of the operations and the parameters are challenged which affect the CQS, like hardness is there or machine speed is there for tablet formulation. Based on the scale up and the scale up outcome, the registration and exhibit batches are manufactured. This is also called as process qualification or validation studies. Sometimes the validation studies are performed on the commercial batches, and sometimes these are performed onto the exhibit batches. Exhibit batches are also the type of validation batches, because the exhibit batches are taken to show that the product can be manufactured in the commercial scale. Then manufacturing in the production scale and the environment. Then report writing. Various reports are required to be generated, like multimedia dissolution report, stability report, process development report, scale up report, and many more. And the last document for the formulation R&D is PDR, that is pharmaceutical product development report. Then filing approval and commercial production. So once you have done with the registration and exhibit batches, you are okay with the bioequivalence studies, and you have the stability data, then the generic product can be filed to the regulatory authorities. Then it will go into the review, and regulatory body may give queries. So generic product manufacturer or applicant will respond to that queries, and based on that the generic product will get approval by the regulatory authorities for the commercialization. Then the batches are manufactured for commercialization. This is also known as or called as launching. Then after launching, the commercial production starts. After that, the life cycle approach comes. Whenever the generic formulation required changes, these changes go into SUPAC and the variation filing. The SUPAC and variation filing can be differing for the different regulatory bodies. For example, site changes, equipment changes, batch size changes. So this is regarding the filing approval and commercial production. So I hope that I have covered the topics related to generic product development. So whenever you are asked a question that what is the generic product development pathway or steps, then you can summarize like that: initial development, scale up exhibit, and filing and query response. So the interviewer will understand that you have the understanding about the generic product development. The critical points in the generic product development are bioequivalence, then meeting to the impurities or meeting the impurity guidelines, then stability, and the robust and scalable manufacturing process. So these are the key requirement of the generic product development. First is bioequivalence, then is stability, and third one is the process of manufacturing. I hope you might have got good understanding. This video got lengthy little bit, but if you go through the video, you will get the understanding about the generic product development. Thank you for watching the video. Please do like, share, subscribe to the Pharma Learning in Depth channel. Thank you.